46, we are talking about the peroxisome. It's a membrane enclosed organelle. So it is surrounded by a membrane. It contains like multiple um, um, it does multiple actions like beta oxidation of the very long chain fatty acids. Very long chain fatty acids. This is strictly peroxismal process, so it's basically occurs only here. And there is also alpha oxidation of a branched fatty acid, not like very long. This time it's branched fatty acids. So strictly also peroxismal process. We also have the catabolism of amino acids and ethanol. It also includes the synthesis of bile acids and the logins, plasmalogens. It's an important membrane phospholipid, especially in the white matter of the brain. So it does create or make some bile acids and also plasmalogs. And it is an important membrane phospholipid, and it's basically found in the white matter of the brain. Let's see. We have, okay, that's okay. For the Zellweger syndrome, it's an autosomal recessive disorder of peroxisome biogenesis. So it's autosomal recessive disorder of peroxisome biogenesis. So basically, like the creation or the synthesis of this peroxisome. Due to mutated PEX gene. That's the name of the gene. So mutated PEX gene ends up being Zellweger syndrome in the patient. It includes hypotonia, seizure, jaundice, craniofacial dysmorphia, hepatomegaly, and early death. Hypotonia, seizure, jaundice, craniofacial dysmorphia, hepatomegaly, and early death. We also have the Refsum disease. In the Refsum disease, it is an autosomal recessive this time of alpha oxidation. So basically, there's a problem within the alpha oxidation. Okay, it causes a buildup of phytanic acid due to inability to degrade it. So we cannot degrade the phytanic acid. That's why it builds up. It causes scaly skin, ataxia, cataracts, or blindness, shortening of fourth toe, epiphyseal dysplasia. Treatment include diet and plasmapheresis. So basically, we are cleaning the plasma from the buildup of the phytanic acid. We also have adrenoleukodystrophy. Adrenoleukodystrophy. It's an X-linked recessive disorder. So basically, it only occurs on the X gene. Disorders of beta oxidation. This time, it's a problem of the beta oxidation due to mutation in the ABCD1 gene. ABCD1 gene. Nice. Is the very long chain fatty acids buildup in adrenal glands. So that's where the name came from. It's a buildup of very long chain fatty acid within the adrenal glands. It's white or in the white or leuco matter of the brain. So it's either in the adrenal or white leuco matter of the brain or in the testes as well. It's a progressive disease that can lead to adrenal crisis, adrenal gland crisis, a progressive loss of neurological function, and death. So basically, it's because it's progressive issue. Let's see what we have over here. So for the peroxisome itself, it's mentioned disorders of peroxisome metabolism. There could be a problem with the very long chain fatty acid and the branched chain fatty acid. That's the phytanic acid. It cannot be metabolized by mitochondria, so transported to peroxisome to undergo special form of oxidation because the very long chain fatty acids and the branched chain fatty acid cannot be metabolized by the mitochondria. That's why we move them to the peroxisome over here. Peroxisome disorders, they include accumulation of very long chain fatty acids in the brain and leading to permanent neurological damage. That's one. For the Zellweger syndrome that we talked about, it's defective peroxisome biogenesis, like a problem within the genesis of biosome, peroxisome itself. It causes craniofacial abnormalities and hepatomegaly. There is an X-linked adrenoleukodystrophy. That's the defective transport of fatty acids, and it presents in childhood with adrenal insufficiency testicular abnormalities. Adrenoleukodystrophy says like it's a defective transport of fatty acids. And basically, it doesn't say over here like what it is specifically, but it says it's a problem with the ABCD1. So I guess that the problem with ABCD1 gene leads to a transportation problem, like a fatty acid transportation problem. So the very long chain fatty acid, special form of beta oxidation, defective in Zellweger syndrome, cerebrohepatorenal syndrome. This disease is due to defective myelin synthesis. Okay, so over here, it's an autosomal recessive disorder produced by biogenesis due to mutation PEX gene, hypotonia, seizure, jaundice, craniofacial dysmorphia, this, and then hepatomegaly and early death. For the branched chain, that's a very long chain. For the branched chain, a branch points at odd number carbons such as phthanic acid by alpha oxidation is defective in Draftsam disease. Results when accumulation of phthanic acid in the body. Treatment is a strict avoidance of chlorophyll in the diet. So as we said, that the Draftsam disease could be treated with diet, and that is strict avoidance of chlorophyll in the diet. That's one. It's actually it's the only thing because the problem was the accumulation of phthalic acid within the body, and it's the problem with the branch points at odd number carbon. What else? We have functions of the peroxisome. Again, it's synthesis and degeneration of hydrogen peroxide. Second, phospholipid exchange. Peroxisome contain enzymes that convert phosphatidyl serine and phosphatidyl thioamine plus bile acid synthesis. Okay, all of these. In addition to our cytoplasmic organelles, they are like the peroxisome, our cytoplasmic organelles containing oxidative enzymes such as catalase, D-amino acid oxidase, and uric acid oxidase. These organelles are ubiquitous among eukaryotics and are most abundant in liver and kidneys, where detoxification of ingested environmental material occurs, and the liver also play a role in the breakdown of fatty acid. So basically, they are mostly abundant in the liver and kidneys because they are two organs that do this detoxification of the toxic material within the body. And the liver also plays a role in the breakdown of fatty acids. So we also have the peroxisome deficiency. What if we don't have enough number of peroxisomes within the body? It's several genetic diseases are associated with impairment or absence of peroxisomes. These patients fail to oxidize very long chain fatty acids and accumulate bile acid precursors. So basically, it's a problem like with the degradation or oxidization of the fat of the very long chain fatty acids, and of course, because we cannot do that, is going to accumulate a lot of bile acid precursors. The four most common disorders are Zellweger syndrome, neonatal adrenoleukodystrophy, infantile Draftsam disease, and hyperpipicolatemia. Hyperpipicolatemia. What a nice name. Okay, so for the proteasome, it's barrel-shaped, okay, so a barrel-shaped protein complex. It's not just one protein, it's a complex proteins. So barrel-shaped protein complex that degrades polyubiquitin tagged proteins. Only one obvious job to do, and that is the degradation of polyubiquitin tagged proteins. So they're proteins that they do have a tag on them, and this tag is called polyubiquitin. So this proteasome plays a role in many cellular processes, including immune response, like compatibility complex 1 mediated. That's thanks to proteasome, and defects in ubiquitin proteasome system also implicated in diverse human diseases, including neurodegenerative diseases. So if we do have a problem with that system is that has two parts: the ubiquitin, that is the tag, and the proteasome that notices the tag, or actually degrades whatever that tag is, like degrades that protein that has such a tag. So if we do have a problem with that, it's implicated in diverse human diseases. So many diseases have this. If we do have this problem, then we may have a wide variety of diseases, including the neurodegenerative diseases. Nice. So it's barrel shaped. It's ATP required to pass through the tubular proteasome. Um, well, regardless of the shape, it seems like we do need some energy. We do need some ATP to pass through the tubular proteasome, because it's like barrel shaped. So we would say it's some kind of tubular. Yeah, that's not nice, tubular, but anyway. Um, Ubiquitin tag. So ATP dependent attachment of ubiquitin. Attachment of four or more ubiquitin proteins are required before proteasome degradation. So the attachment of this tag to this protein requires ATP, and we need at least like four or more tags. Four or more ubiquitin proteins are required before the proteasome um, uh, degradation, before the proteasome starts degrading that material. We also have the Parkinson disease. It doesn't say anything in 2003 about the Parkinson, but let's see. There is a defect in PARC, PINK1, and DJ1 genes are responsible for autosomal recessive Parkinson disease that have an early age of onset below 50 years. In Alzheimer's disease, the defect also plays a role. So if we have someone who has Parkinson and that's like an early onset, like before the age of 50, that may be the defect due to pink gene, pink 1 or DJ1. So the common here is number 1. And we also could find this problem in Alzheimer's disease. Okay, that's for the proteasome. We also had proteasome that helps with the matrix complex 1, mainly degrade intracellular proteins, while lysosomes that are like involved with the matrix complex 2, they do degrade extracellular proteins. So proteasome works with the matrix compatibility complex 1, and it mainly degrades intracellular proteins like within the cell, and the lysosome, it works with the major histocompatibility complex 2, which degrades the extracellular proteins. Let's see what else we have here. We do have the cytoskeletal elements. Doesn't have a lot of notes, but basically it's a network of protein fibers within the cytoplasm. So it's a network, not like a single one, like multiple of those protein fibers within the cytoplasm, not nucleus, not mitochondria, like within the cytoplasm, but still within the cell, that supports cell structure. Like they do give support, cell and organelle movement, they work with that, and cell division. So they do support cell structure, cell and organelle movement, and cell division. Okay, a lot of great jobs. Type of filaments, we have predominant function and we have examples. So these filaments could be of different type. We could have microfilaments or intermediate filaments or microtubules. For the microfilaments, they are required, like they do function in the muscle contraction and cytokinesis. The examples are actin and microvilli. So muscle contraction, it could be actin or microvilli, and also in cytokinesis. For the intermediate filaments, they maintain cell structure, just like vimentin, desmin, cytokeratin, lamines, glial fibrillary acid proteins, and neurofilaments. Okay, all of these are intermediate proteins, and because they're intermediate, they do maintain the cell structure. We also have the macrotubules. They are required for movement. Okay, movement and cell division. Movement and cell division. They're the cilia, flagella, mitotic spindle, axonal trafficking, and centrioles. All of these are involved in movement and cell division. So, let's see, what does it have about the desmin? The desmin is an intermediate tubule. It supports the cell structure. It's a smooth muscle actin and called desmone are also used. So within the small muscle, within the small within the smooth muscle, we could have desmin or actin or called desmone. Okay. We also have cytokeratin. It's made up of glycine and alanine because of their small structure, are tightly coiled and form large number of hydrogen bonds. The cytokeratin is basically made up of glycine and alanine. Why? Because their small structure are tightly coiled and they do form large numbers of hydrogen bonds. Um, Additionally, has significant number of sulfur-containing amino acids, cysteine, and formation of disulfide bond with bond, it should be bond, right? Disulfide bond between cysteine residues that gives additional rigidity and toughness to the protein. Okay, a lot of info. Made of glycine and alanine. Because of their small structure, they are tightly coiled and form large number of hydrogen bonds. Additionally, has significant number of sulfur-containing amino acids, cysteine, and formation of disulfide bond between cysteine residues that give additional rigidity and toughness to the protein. Okay. So for the microtubule, okay, and the microtubule was, as it's supposed to say over here, it aids in the movement and cell division, okay. So for the microtubule, it's a cylindrical outer structure composed of a helical array of polymerized heterodimers of alpha and beta tubulin, okay. So we do have like the very smallest part of it is a heterodimer, as it says over here, heterodimer, like two pieces together, this blue thing, this green thing, okay. So a heterodimer is of what? Of alpha and beta tubulin. So this thing, one of them is alpha, okay, the other one is beta. These heterodimers are polymerized, okay? Polymerized in a way uh, like in a helical array. So it's just like a helix, just like this shape. All over, like helical array keeps looping, it ends up as cylindrical outer structure. It ends up as a cylindrical structure, just like this one. So heterodimers of alpha and beta tubulins, they are polymerized into helical array, ending up as cylindrical body, and does have a positive end, like with a positive charge over here, and have a negative charge, okay. Within like this whole row of alpha and beta tubulins, it's protofilament, this is what it's called. So each dimer, each one of those that has two parts, has two GTP bound, like each dimer of those has two GTP bound to it, incorporated into flagella, cilia, mitotic spindles. So all of that, like that has two GTP to each one of those heterodimers, two GTP is incorporated in the flagella. It's used within the flagella, within the cilia, mitotic spindles. Also involved in slow exoplasmic transporting neurons, For molecular motor proteins, transport cellular cargo towards opposite end of the microtubule. So we do have like within the molecule some motor proteins that they are the transporter proteins. They do transport the stuff like the cellular cargo. The cellular cargo is like the content or the produce that should be moved from one place to another. So the cellular cargo towards the opposite end of microtubules. So we do have like microtubules over here, and we need to move like one thing from over here to the other part. So all over the microtubules, we do have like some molecular motor proteins that do that and move this cargo. So if it's a retrograde to the microtubule, like the if it's backwards movement, like retrograde to microtubule, like from the positive end of the microtubule to the negative end, is called dynein. But if it's anterograde to the microtubule, like it's from the negative end, from over here to the other way, like anterograde this way, then it's kinesin. So all of these dynein and kinesin are molecular motor proteins, and they do move. The dynein moves from the positive end to the negative end, and then the kinesin moves from the negative end to the positive end. For the Clostridium tetani toxin, or poliovirus or rabies virus and herpes simplex virus, they do use dynein for retrograde transport to the neural cell body. They do use dynein, okay? And the dynein should move stuff from the positive end to the negative end, okay? So this is the positive end to the negative end. So what uses that is the Clostridium tetani toxin, poliovirus, rabies virus, and herpes simplex virus for retrograde. Oh, it was retrograde. Yeah, retrograde. Did we say that? Okay. So uses dynein for retrograde transport to the neuronal cell body. The herpes simplex virus reactivation occurs in anterograde transport, while only the herpes simplex virus reactivation. So if it's first encounter with the herpes simplex virus, then the virus uses dynein to, go up to the like from the end of the nerve to the ganglion of the nerve. Okay, so it ends up here. So this way it's retrograde. That's why it uses dynein. But if it's reactivated and needs to, like after stress or illness, then it will move with use the kinesin. Its reactivation occurs in the anterograde transport from cell body, like from all over here to the skin. It's a kinesin mediated. So slow anterograde transport rate limiting step, peripheral nerve regeneration after injury. For the slow anterograde, so if it's anterograde, if it's this way, then it's slow, okay? It's a rate limit step of peripheral nerve regeneration after injury. Drugs that act on microtubule, this is just to memorize it. Microtubules get concentrated very terribly. So we have mebendazole, okay, and that's antihelminthic. We have rhizofulvin, that's antifungal. Colchicine, that's anti-gout. And we have vinca alkaloids, they are anti-cancer. And taxans, they are anti-cancers. So negative end near nucleus, positive end points the periphery. This is just to know where the negative end and where the positive end. The negative end is near the nucleus, so it's close to the nucleus over here. And we have negative end over here, while the positive end is like close to the periphery of the cells. Okay, ready, attack. That's just for memorization. Let's see what we have over here. So it says that kinesin is microtubule associated motor proteins. They transport towards the plus end of the microtubules, okay? Or positive end, rapidly growing far from nucleus, anterograde transport. It uses ATP as a source of energy. So basically just what we talked about. And that's it for now.